Hi, in this video, we're going to cover viral life cycles, and notice that the word life is in quotations because, as we've talked about before, viruses are not technically living things, but they do affect living things, and that's why we're talking about them. And so they don't necessarily have a life cycle, but they do have this infectious cycle of how they kind of make more of themselves. And so that's why we're putting the word life in quotations. So let's start with bacteriophages. Bacteriophages are viruses that specifically kill and infect bacteria. So their host is specifically a bacteria. And so here's our little bacteriophage is going to bind to the surface of the bacteria using its tail fibers, and then it's going to inject its DNA or RNA, depending on the type of bacteriophage, but let's just assume this one is a DNA bacteriophage. It's going to inject its DNA into the host. And so in this diagram, they have the bacteriophage injecting its phage DNA into the host bacteria. So this big blob right here is the bacteria, and the phage DNA is in blue. Okay, and just to keep things more clear, in this diagram, they're also showing the host, the bacteria's DNA, shown in pink. So yeah, so the phage injects its DNA into its host, which again is a bacteria. It could be E. coli, it could be Salmonella, depends on the bacteria. And then that DNA that gets injected into the bacteria is used to make more phages, because remember, DNA carries information, and the information that is carried is essentially the recipe to make more phages. Now you're probably wondering, why would the bacteria make more phages? Well, that's basically how viruses work. They trick their host into making more phages. It's kind of like if I went to a McDonald's and in the cookbook, I'm not really sure how it works, but I'm assuming that in the restaurant there's like a cookbook or something. I slip in the recipe to make a Wendy's burger, right? Because I'm pro Wendy's, not McDonald's. Well, actually, I like both, but whatever. So imagine I go into McDonald's and I sneak in a Wendy's recipe to make their Wendy's burger. And the workers don't realize, the people working at McDonald's don't realize that they're following the Wendy's recipe instead of the McDonald's one. That's kind of what a virus does. It sneaks in its recipe into the host, and the host doesn't know any better. It makes more viruses. And these viruses assemble, so all of the little pieces of the virus are made. So, you know, the McDonald's workers are making the Wendy's burger patty, which is square, and then they're like making the cheese and the buns, and then the whole hamburger gets assembled, and then now McDonald's is selling Wendy's burgers, right? So that's basically what's happening. And here the bacteria explodes with all of these viruses. It does kill the host. So because the bacteria explodes with, here they're showing like 10 or 8 bacteriophages, but in real life, there's probably like 1,000 or even more bacteriophages that explode out of the host cell. And the bacteria dies in this process. So there's this type of kind of life cycle for a virus. Now, some bacteriophages are a little bit more sneaky, where they keep their host alive, which is kind of scary. So the first part of the process is the same. The bacteriophage injects its DNA into the bacteria, so the host. But this time, that DNA, the recipe to make a phage, is incorporated into the host DNA. So this is not just me sneaking in the recipe into Wendy's. I am sneaking it into the actual McDonald's cookbook. So I'm sneaking into McDonald's. I have my Wendy's recipe. I'm going to literally shove it into the cookbook so that it looks like it's part of the McDonald's cookbook, right? So I'm hiding it in there. And that is what the bacteriophage does, is it hides itself. Now, when this DNA is hidden in the bacteria's DNA, it's called a prophage. Anyway, then the bacteria just kind of live its life. So the bacteria, continues to grow and multiply, but as it replicates, it is, unintentionally replicating the phage DNA as well, so the virus's DNA. Now, at one point or another, there's some kind of stressful situation, something that triggers this phage DNA to then separate. So as you can see, the blue DNA is now separated. and then we go back into the normal making more phages, and then the bacteria gets lysed after. And so this is another option for phages. So they can either go through this cycle, which we call the lytic cycle, meaning that it kills the host immediately. So lysis means to explode. So this is the lytic cycle. And then here in this cycle, we are hiding for a little bit first, and we call this the lysogenic cycle. Okay, now animal viruses go through a very similar process, and so you're going to see a lot of similarities here. Viruses are going to attach to their host, and again, these are animal viruses. So, for example, something that infects humans or something that infects pigs or birds, whatever kind of virus we're talking about. The virus is going to bind to its host, and then the entire virus goes inside. So the main difference here is that the whole virus enters rather than injecting its DNA or RNA. So the whole virus enters the host cell, and then it releases its genetic material, whether it's DNA or RNA. That genetic material usually goes to the nucleus or is used directly in the cytoplasm, but regardless, we're making more viruses here. And again, the host is being tricked into making more viruses. And the virus assembles and viruses come out. Sometimes this kills the host cell, sometimes it doesn't. Most of the time it does kill the host cell though. And so new viruses are released, and those new viruses can go on and infect another cell. Another thing I wanted to mention is that the host for a virus is very specific. So for example, this is influenza. This is human influenza, which will infect only humans, and not just that, it will only infect the epithelial cells that are found in the lungs and respiratory system. So when you get the flu, you have symptoms like sneezing, coughing. It's all respiratory, right? And that's because influenza primarily infects the cells that are found in your lungs. So not only are viruses specific to the type of animal or host, but they're also specific to the location on a host if you're talking about an animal, for example. One other thing I wanted to mention about the life cycle of viruses is that there is a small dilemma when it comes to RNA viruses, and that is whether the RNA is the positive or negative sense RNA. So positive sense RNA essentially means that that information can be directly used to make new viruses. So there's no issue when it comes to positive sense RNA viruses. But negative sense RNA is basically inverted. So the information to make a new virus, imagine if it was written backwards, like if you wrote your name backwards, right? And so that information needs to be written in the normal direction first. So basically it needs to be decoded in the normal direction before it can be used. And so there is this enzyme called RDRP, the viral RNA-dependent RNA polymerase, which basically converts it from the negative to positive. And then it can be used to make more viral proteins and viral components. Now, by the way, this RDRP is usually provided by the virus that is causing the infection. So if you are a negative sense RNA virus, you better have your own RDRP, or then you wouldn't be able to do anything because all of your information is written backwards. So you basically bring this enzyme that rewrites your information in the forward direction. All right, so that was kind of a small intro to viral life cycles. Now, I was just doing very general viral life cycles. There are some specific viruses that have very complex life cycles. So we saw influenza, very straightforward, but for example, here, this is the HIV life cycle. There's a lot more steps. There's a lot more, proteins that are involved. And so that would be for another video, but hopefully this video was a good intro to just general viral life cycles.